My name is Martín Soñora. I am a PhD student. And first of all, I would like to thank the organizing committee for giving me the opportunity to be here. This work was carried out at the Biomolecular Simulation Group of the Institut Pasteur Montevideo under the supervision of my advisor, Sergio Pantano. And due to lack of time, I will be very brief in my explanation. So let's start. Okay. Flaviviruses share a common structure composed of a capsid inner core surrounding the genomic RNA. And these viruses contain a lipid membrane, and embedded in their bilayer, there are 180 copies of envelope and membrane viral proteins arranged in an icosahedral symmetry, taking a T=3 like organization of sulfide heterodimers. And technological advances on computers and experimental techniques such as cryo-electron microscopy are allowing us to simulate and analyze progressively more realistic and huge biological systems. In fact, the present capability to simulate this kind of systems may be further enhanced by the use of multiscale strategies, which combine atomic resolutions with coarse-grained resolutions. But yet, building and setting up intricate viral systems like viral-like particles present here remain a computational challenge, in particular because the experimental resolution and the distribution of different constituents in confined compartments, like a small space between viral proteins and their membranes, may hinder the proper setup, equilibration, and reproducibility of the simulations. Today, cryo are achieving high resolution structures of multi-protein complexes, for example, such as capsids and envelopes. However, membrane components are still hard to solve with enough atomic resolutions for the accurate generation of molecular models, for example. So the only thing that we know with confidence about viral-like particles is the position of the atomic coordinate of the viral proteins. We don't know anything about the atomic position of the viral membrane lipids, for example. And in the best case scenario, only raw spatial information about geometric boundaries and lipids densities of membranes can be obtained. Like this one here, for example, this section here corresponds to the inner leaflet of the viral membrane. And as you know, computationally speaking, the membrane packaging problem is an NP-hard problem. So in this regard, we identify three main problems for the multiscale modeling of this kind of systems. Building accurate protein membrane models according to experimental restraint in confident conditions, generating a multiscale representation of the systems, both first and second. Remember that we don't know anything about lipids and water molecule coordinate position from this kind of biological system. That we only know is the protein atoms coordinate position from the cryo density maps. And the third issue is having a simple and robust protocol for equilibrating and simulating the multiscale system. And this is achieved with the SIRAH force field. And SIRAH is a coarse-grained force field developed in our laboratory. And in the context of molecular dynamic simulation, the term force fields refers to the combination of a mathematical formula and associated parameters that are used to describe the energy of the molecules as a function of its atomic coordinates to explore, for example, intra and intermolecular interactions. Well, to solve the packaging problem, we use the Packmol software. So what were the Packmol's improvements? This software creates an initial point for molecular dynamic simulation And this is done by the packaging molecules in a defined region of the space. With Packmol, different types of spatial constraints can be attributed to the molecules or atoms. And in this way, Packmol makes it easy to create ordered systems such as lamellar, spherical, or tubular lipid layers. And in your case, as Flaviviruses has an icosahedral structure, we will use the configuration of a perfect sphere because it's the closest geometrical figure to an icosahedron. And here we present a multiscale scheme, and this implies the, the coexistence of different granularities or resolutions at the same time in the simulation of the system. So because of the heterogeneity of components such as protein, lipids, solvent, ions, etc., that constitute this type of systems, and because of the variety of molecular elements may vary widely between the different components, therefore, this directly involves in the customization of the short range packaging function. So imagine that at the beginning of the packaging process, there is a lot of space inside the simulation box. And it is easy to place molecules in those conditions. But as the molecules are placed during the packaging process, less and less space is left and clashes start to appear. In this regard, we introduce a final penalty score corresponding to the sum of the default and short range terms, solving very good the packaging problem, the packaging problem. So this kind of packaging guarantees that short range repulsive interaction don't disturb the simulations. This scheme here represents the pipeline for building and simulating enveloped BLP systems at multiscale resolution. In left panel, I am showing the stepwise protocol for calibrating enveloped BLP. The calibration is split into protein BLP and membrane protocols, and inside each step, there are several energy minimizations, equilibrations, and short production steps with restraints, resolution steps to fix, for example, density issues. This is an iterative process to correct, for example, density issues, and then after resolving the system, when problems were solved, the system is run for several microseconds. And in right panel, I show the resulting enveloped BLP of Zika virus after executing the pipeline. And the molecular components of the system are shown here as an onion-like shape, showing each component, the protein, membrane, lipids, and the solvent too. Well, then I directly compare the density maps measured during the simulation, named here protein MD and membrane MD, pink and green respectively, and compared to the electron density map obtained from the cryo electron microscope experiment, named here experimental, W1. Seen from figure, the matching between density maps is outstanding. The excellent agreement extends to the protein part and the bilayer, which shows a flattening in the region away from the transmembrane helices. Here, this size. To acquire a quantitative, okay, thank you. To acquire a quantitative assessment of the quality of the model, I calculate the global correlation between the density maps of the simulated BLP and the cryo MD data. And the initial correlation coefficients of the entire BLP results as high as 96% at the experimental resolutions, and temperature and breathing effects drive the VLP away from the perfect symmetry, progressively reducing the correlation along the simulation. Nevertheless, our simulation continue to reflect the experimental data to a large extent, as the correlation for the Google VLP remain over 90%. Conversely, calculating the correlation only on the membrane shows a marked increase from the initial conformation. Remember that initial conformation is a perfect sphere from Packmol software, and the presence of the protein envelope impose significant distortion to the lipid bilayer. And as a result, the spherical Packmol packaging solution produce better match in the neighborhood near the transmembrane helix, which match with the radius used to place the lipids with Packmol software. In contrast, the bilayer regions close to the 5-fold axis here, which are flatter in the cryo maps, show significant mismatch, as you can see here in the orange bits and the blue density. However, the combination of an appropriate force fields like SIRAH force field with a robust equilibration protocol rapidly fix these small issues. Such an agreement with experimental data provides a validation of the building protocol and the multiscale simulation to explore the dynamics of the VLPs. In this slide, you can see global molecular dynamics descriptors. At left, you see structural features of the VLP along the simulation. You can see, for example, radius of gyration, root mean square deviation values of each protein components in the simulated system. Here values are reported for the entire VLP, envelope plus membrane proteins, and also averages over individual monomers. The dashed line here corresponds to the experimental radius of gyration of the VLP. As you can see, we find a very good agreement with experimental data. In the right panel, you see the normalized B factor of envelope and membrane proteins, and the correspondence of values from experimental and simulated structures at transmembrane regions of envelope and membrane proteins is notorious, as you can see in these regions. Pointing to a good quality of the protein membrane model. Moreover, the flexibility at regions triggering, for example, the infection process and the immune escape, such as the fusion loop here in green and the variable glycosylation loop in cyan, is very well reproduced too. Well, as a home take message, we develop a robust methodology for assembly of naked and enveloped viruses. We present equilibration protocols, provides a system ready for production molecular dynamic simulation within a few days. Our method is transferable to multiscale, all atoms, coarse grain, super coarse grain models, and more important too, that we get high correlation with the cryo-electron microscopy experimental data that validate very well our model. Thank you very much for your attention, and I will be happy to answer your questions.
That was a wonderful talk. Thank you so much.
Thank you.
So we have like a minute left, but if there are any very quick questions, maybe we can take one, but if not, perhaps it would be good to answer those questions in the chat and then move on to the next speaker.